The surprising bottleneck in living materials is not whether biology can do something useful, it is whether we can manufacture it, measure it, contain it, certify it, repair it, and then retire it with industrial repeatability. Living and biosynthetic materials are a broad family, not a single invention. They blend cells, proteins, virus templates, or cell-linked polymer processes. The goal is to make materials that sense their environment, change shape, grow, or repair themselves. That collides with a core industrial assumption. Most materials are inert after fabrication. They keep a fixed geometry, and they only get worse with time. A practical insight is that the field is really a stack of material classes. Bio-derived materials sit on one end. Bio-enabled and bio-hybrid materials sit in the middle. Engineered living materials sit on the far end, with the highest containment and life cycle demands. This is not academic. It determines whether you test something like an inert product, an adaptive product, or a contained biological system. It also shapes regulation, insurance, and the failure modes that matter. Several technical routes are already established. Protein modification has become a usable toolkit for tuning properties and getting smarter behavior. Inorganic protein hybrid structures are also a clear class, aiming to combine biological recognition and self-assembly with inorganic strength, optics, or electronics. Viruses can be used as templates too, not as pathogens, but as repeatable platforms for nanoscale organization. Polymer-based nanocomposite particles remain a workhorse approach, embedding nanoscale function inside polymer matrices for bio-oriented uses. If you care about sensing and computation at the edge, there is a key bridge. Protein-integrated electronic nanodevices try to pair biological selectivity with electronic readout and control. The idea is simple and hard. Signal fidelity and mechanical integrity have to survive real environments, not clean lab conditions. Now for the part that gets underestimated. Interfaces are the battleground. Protein inorganic, protein electronic, and polymer nanocomposite interfaces can fail under mechanical cycling, thermal swings, hydration changes, chemical exposure, and simple time. A demo can look perfect on day one. Then the interface delaminates, drifts, or degrades quietly over weeks. Interface reliability is often the practical gate between prototypes and certified devices. Human contact applications raise the stakes. Host response is not a late-stage checkbox. If a material sits near tissue, compatibility and long-duration performance drift become core metrics. The material is not only trying to work, it is trying to work while the body, or a tissue-like environment, actively reacts to it. A newer and provocative concept is living synthetic polymerizations. Here, polymerization is coupled to living systems. That forces containment, growth control, and life cycle governance into the design from the start. It also changes what maintenance means. Instead of patching cracks in a passive surface, you may be managing viability, nutrient dependence, shutdown triggers, and controlled decay. Manufacturing reproducibility is the other major constraint. Biomolecular and living components vary with environment, expression conditions, handling, and degradation state. That variability turns into batch consistency risk, warranty ambiguity, and slower approvals. For inert materials, a single-point test can sometimes stand in for a lifetime. For adaptive or living materials, it cannot. You need time series measurement, drift tracking, failure rate data, and disposal outcomes that are as measurable as performance. containment and disposal deserve the same rigor as performance. Any system that retains viable function after deployment has to specify boundaries, shutdown methods, replacement intervals, and waste streams. Without that, the risk shifts from the material breaks to the material changes state outside the expected envelope. That includes uncontrolled growth, contamination, false sensing, and persistence beyond intended boundaries. At scale, the danger is not only technical failure, it is unaccountable living components embedded into buildings, consumer products, medical interfaces, or ecosystems. Time horizons matter because they shape which pilots are sensible. From 2026 to 2050, expect concentration in labs, controlled coatings, sensors, biomedical adjacent interfaces, and bounded trials where replacement is easy. From 2050 to 2100, certified adaptive surfaces and repair-capable composites become plausible, but only if standards mature around drift, containment, disposal, and interface reliability. Further out, living architecture and infrastructure-adjacent deployments remain possible, yet still gated by governance, waste handling, and long-duration qualification. One institutional signal is clear. Major funding systems are still placing bets on the materials frontier. The point is not any single project. It is that the pipeline is being kept alive, which means leaders will be expected to separate science wins from deployment readiness. So what should a senior leader do now? Force classification upfront before money moves. Fund metrology and life cycle testing before scale-up. Treat containment as a design requirement, not a compliance afterthought.